Welcome back. So here we are. So in these slides, I'm just going to show you what I normally draw on the board, but I think this is better. If you, well, you are watching these videos, and that's the right thing to do because the following slides are going to be a mess of a jumble if you don't see them animated. So I'm going to turn on my slideshow here and try to resize. Get it in the window. Okay. Cool. Here we go. So this is what I call a DNA sequence upstream of a gene. So this is before the gene. If you start here and you're RNA polymerase, you go to the right. So you're always going to go towards this 3 apostrophe. That's 3 prime. And if you don't know why, don't worry about that at all. I haven't talked about it. But we're always going to go towards the 3 prime. And so the gene is off this way. ... here means this molecule of DNA continues in this direction, and it continues in this direction. So this is one DNA strand. It is one molecule, one very long chain. And there is another long strand of DNA base paired to it. So they are not covalently bonded to each other, but they are tightly held by a huge number of weak bonds, and the bonds are between each pair of nucleotides that perfectly base pairs. You'll see that the opposite strand goes the other direction. The 3' goes the other way, and that's what it's set up as. So review base pairing if you've forgotten it. And then what we're going to look at here is the sigma factor. Let's look at sigma factor. So remember I showed you this sequence, TTGA and then TATAT. That's the sequence the sigma factor is looking for. And then what we're going to see is starting 7 bases later, that's the beginning of the gene. That's where the messenger RNA is going to actually start. So we're going to see the messenger RNA that just matches this sequence. And it's going to start here and go to the right. But before that happens, RNA polymerase, which is going to make that messenger RNA, it needs to find this. And it can't. What can find this is the sigma factor. And so the sigma factor is going to look for this sequence, which is called the promoter, or the promoter region. So the sigma factor is going to look for the promoter region, and once it finds it, it sticks. It's a very happy sigma factor. It found what it's looking for. And now its job is to grab RNA polymerase and set up RNA polymerase on this DNA strand. And RNA polymerase will then look for this spot relative to the sigma factor. And it works very well. Now, RNA polymerase is much, much bigger than a sigma factor. RNA polymerase would be like giant compared to the DNA. That's not that important. So RNA polymerase is going to start making this messenger RNA, and it's going to keep going in that direction. Remember, a gene might be 1,500 nucleotides long. Easily. It could be 3,000 or 5,000 nucleotides long. So we're never going to write the entire thing on a whiteboard or whatever. So DNA, double-stranded, base paired from one strand to the other. RNA is single-stranded. There's no need to base pair this. Okay, let's keep going. So we're going to use the codon table. I used the codon table to set up these slides, and you can verify everything using this table. So let's look at it. So here we are, the DNA sequence at the start of a gene. So we're no longer upstream of the gene, now we're at the start of the gene, and we're going to see what signals are at the start of the gene. Again, we're going to bring in our base paired opposite strand, which goes in the opposite direction. And now I'm going to light up the signals that we could see here. It's hard to see, but these are bold because these are important. This is important, so it's underlined. And then these groups of 3, I've used this kind of shading to show you this is the first group of 3. That's a codon. It encodes an amino acid. That's the second group, third group, fourth group, fifth group, sixth group. They keep going off to the right. If you notice the base pairing ones, they just don't have any clear relationship to these. So each individual nucleotide correctly base pairs, but the pattern you find in this strand really has no relationship to this pattern up here. Now, we could look at this strand and from it know exactly what this sequence is, but that's the only value we have from this strand. So what is this AGGA? Well, this is the ribosome binding site. This tells the ribosome where to go. Now, the ribosome is not going to bind to DNA. The ribosome binds to a messenger RNA, so we're going to have to put that down here. So, remember in the last slide, RNA polymerase was making that messenger RNA, so it continues making that messenger RNA. And so now I've made it easier to see the ribosome binding site. And then this AUG is called the start codon. This tells the ribosome, start here, and the very next 3 are going to be the first codon that means an amino acid. So ribosome binds there at the ribosome binding site, start codon, and then each group of 3 represents an amino acid. And if we go back to the codon table, we can figure out these 3 mean asparagine, these 3 mean valine, these 3 mean histidine, these 3 make threonine, these mean serine. So the ribosome finds this start codon, and then it just starts adding nucleotides one at a time, making this chain. And one interesting point is that AUG is the start codon, but inside a gene it means an amino acid. So it doesn't have any like start meaning. It's just another amino acid once translation has started. So that's the start of it, the start of the gene. These are the codons that make the actual amino acids. This is the protein amino acid sequence. The ribosome binding site is necessary to show the ribosome where the gene is going to be, and once it has bound in this area, it looks for this start codon. If it bound in the wrong place, it would think this is the start codon. So it needs to know, attach here. Okay, so RNA polymerase keeps going, making this messenger RNA. Ribosomes keep running along this messenger RNA, making a protein. And so here we are within the gene. We're not at the start or the end. And again, codon, 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 just groups of 3. And base paired DNA that we ignore. And the messenger RNA keeps getting made by that RNA polymerase, and it matches the DNA sequence. And the ribosome keeps making the protein, amino acid after amino acid. Something I want to show you here is why the start codon is so important. The start codon defines the frame. Okay, so it defines the frame. It says, well, this GCU, that's a codon, and this CGC is a codon. This UCC is a codon. But see, it could have defined it differently. It could have said CCG, and then CUC, and then GCU. If the start codon had been in a different place, those would be the codons. So the start codon defines which of those options is the actual group of codons, and it's really important. So here what I've done is show you what would happen if the start codon had shifted them all to the right so that CCG is the first codon in this part of the gene, and then CUC, and off we would go. And so I've highlighted them as the new codons, and you'll see, they're this and then this. They're just one nucleotide off, but they encode completely different amino acids. So I looked through this because I randomly generated these sequences. They don't mean anything. But I look at these nucleotides, and serine is polar, Alanine is nonpolar. Arginine has a positive charge. Lysine has a positive charge. Serine, serine, serine. So polar, 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 nonpolar in a weird place. And then two positive charges and then another positive charge. So 8 amino acids with 3 positive charges. That is a really big deal. There must be some reason for all those positive charges. Well, if you shift the frame over, we get proline, leucine, alanine, glutamine, serine, proline, histidine. None of these have a positive charge until you get to this histidine. So you've lost 2 positive charges. Also, proline, there are 2 prolines in here. Proline forces a bend in the protein, a bend between the amino acids. There is no bend up here, so this has a different 3-dimensional structure. Definitely different 3-dimensional structure. So the point I'm trying to make here is that if the frame changes, and if instead of seeing UCC as a codon, it starts to see CCG, that randomizes the entire protein, and there's no relationship between this amino acid sequence and this amino acid sequence. So if we go back, this start codon absolutely defines the frame. And the ribosome binding site doesn't. There could be 15 nucleotides in between them, or 10, or 5. But this start codon absolutely defines the frame. Okay, that's that. And then we go downstream of the gene, or really to the end of a gene. So what are we looking at here? Codons, and then they stop for some reason. And so this is really the end of the protein would be here. And we'll see that in a second. And then these nucleotides represent a transcription terminator. So this is the start of a transcription terminator. So again, base paired DNA. And again, our messenger RNA gets made, but why does it stop here? It doesn't say ... It just ends with this 3. Well, it ends at the transcription terminator. So we could look at this DNA and know where the messenger RNA is going to end. And we can look at the messenger RNA and know where the protein is going to end. See, that, UAA, that's a stop codon. That means the protein ends here, no more amino acids. So the last amino acids were serine, asparagine, serine. That's it. Serine is the last amino acid. And you can find that here. TAA, that's a stop codon. That's what it looks like in DNA with a T instead of the U. So if once you know the frame, you can find the stop codon, and that's the end of the protein. And then you look a little bit farther and you find the end of the messenger RNA. And now you've seen all the signals. That's all the stuff you would look for in a DNA sequence. I know it's a lot to take in here, and if you were at all shaky on RNA polymerase and DNA and transcription and translation, then this is hard. But it's worth working this out, because this is giving you more and more a picture of what cells do. And microbiology is about individual cells. And human physiology really depends on individual cells and what they do. And part of what we're learning in microbiology, you can apply to human cells. And we're going to see more and more of that as we get into farther into Unit 2. So thanks for your attention to this video. I hope it's helpful. Have a great day.